Hello friends. We are going to begin first with general pathology, and general pathology is one of that portion of pathology which is extremely important not only for this particular subject but is then going to correlate with all the other 18 subjects that you have. So this you can say is your foundation or is your basic. For pathology, yes, but definitely for medicine, for microbiology and pharmacology at least. So it is very important that you study these particular chapters, that is cell injury, inflammation, neoplasia, immunity and genetics. These 5 chapters are the building blocks for a lot of other subjects that you have. We are going to take them up in the same manner, and with every chapter, not only will we be discussing the high yield points for every exam, not only will we make it simpler for all the students who are preparing or are appearing in their professional examination, but we are also going to focus on something that is often neglected and very much feared by the students, and that is the images. So as we go through the subject, we are going to study a lot of images together, label the images, revise the images time and again so that we can have a better grasp over the subject. So by the end of the subject and by the end of these lectures, hopefully everything will not remain pink and blue and something will definitely add on and start making sense in this particular subject. So let's try to make it as interesting as possible. And the first chapter of general pathology that we are going to begin with is going to be cell injury. Now, before I go on to the different types of cell injury and what is happening, there are 4 questions that require attention. Question number 1. Question number 1 is, what is the most common cause of cell injury? What is the most common cause? And I'm sure many of you already know this. The answer to this is hypoxia. Hypoxia is the most common cause of cell injury. Now from this only, the examiner will ask you the second question. What is the most common cause of hypoxia? So now from the first one only, the examiner has put up a second question. So what is hypoxia? Hypoxia is a decrease in the oxygen concentration at the tissue level. You already know this. Hypoxia is a decrease in the oxygen concentration at the tissue level. Now what is the most common cause of hypoxia? And that is going to be ischemia. So answer to this question is ischemia. That is when the blood supply to an organ is going to go down. When the blood supply goes down, automatically the oxygen supply to that organ will reduce. So the most common cause of cell injury is hypoxia, and the most common cause of hypoxia in turn is ischemia. The next set of 2 questions that we have, the next set is, which is the cell or which is the organ which is going to be most sensitive to hypoxia? Most sensitive to hypoxia. And the answer to that is going to be neurons or the brain. Let's answer this. This is going to be the neurons or the brain. It is said that they cannot tolerate hypoxia for more than 2 to 4 minutes. Beyond this, the neurons will undergo death. Now, make this a habit, students, that whenever you come across a question which is most susceptible or most common, you must automatically, the next question that should come to your mind is, okay, which is the least susceptible or which is the least common? So you should always try and cover both ends of a spectrum whenever you're studying. So if I'm saying that the cell which is most sensitive to hypoxia is the neurons or is the brain, you should automatically ask me which is the cell which is least sensitive to hypoxia. So now going to question number 4, which is the cell which is least sensitive? And students, if I'm saying least sensitive, can I safely also call this the most resistant? This becomes the most resistant to hypoxia. And now we are expecting this question in the future exams, and the answer to this question is fibroblast. The answer to this question is fibroblast. So we've done, before starting into the intricate details of cell injury, we've done 4 main questions. What is the first question? The most common cause of cell injury is hypoxia. The most common cause of hypoxia is ischemia. The cell most sensitive becomes the neuron, and the cell least sensitive or most resistant becomes the fibroblast. Now, if this particular set of MCQs are clear, now let's understand what exactly is happening in cell injury. So beginning with a basic flowchart, students, and then I'll go into the details. Whenever there is any injurious agent, see now an injurious agent can be a physical agent like heat, cold. It can be a chemical like poison, toxin, alcohol. So there are a varied number of injurious agents. Anything and everything can act as an injury to our body. But whenever something tries to harm your body, whenever there is an injurious agent present, remember this injurious agent will come to your body. But will your body just give up? No. The first step is cellular adaptation. Your body really wants to protect you till the very end, and your body will try to adapt at the first go with whatever it faces. But the problem arises that if this injurious agent is very severe, or if for many days this injurious agent keeps on poking your body and keeps on trying to injure your body, there will come a time when your body will give up. And that is when it enters into reversible cell injury. Now, still the body has kept it as a zone that okay, cell injury has started, but even now if this injurious agent goes back or is withdrawn, whatever changes took place were reversible. The cell will still come back to its normal state. So still the body is trying to give you some time. But the problem arises is that if still the injurious agent continues, we are now entering into irreversible cell injury. So now you can see the basic flowchart says that we will first try to adapt. If we fail to adapt, we have entered into reversible cell injury. And if it further continues, we enter into irreversible cell injury. And students, can I safely say that this would be cell death? Once the cell cannot come back to its original state, it will ultimately progress to cell death. Now, what are the different types of cell death that you are aware of? I'm sure ever since you've entered second year and you've started studying pathology, these are the 2 types of cell death that you've heard of. We've got necrosis, we've got apoptosis. Now I'm going to tell you some newer forms and make it a habit that whatever is recent, whatever is new, will automatically, there are more chances of it coming in the exam. The examiners will try to test your knowledge on those. The newer forms of cell death include, all that you need to do, combine necrosis plus apoptosis. I give it a new term, that is necroptosis. The 4th one is again a newer one, pyroptosis. And the 5th one is something that not many books cover, that is referred to as anoikis. So now students, we have the different types of cell deaths: necrosis, apoptosis, necroptosis, pyroptosis and anoikis. Not only is this particular flow diagram very important for your basic knowledge, but this also is going to give you an outline of how we are going to deal with this chapter. So first we'll start with reversible cell injury. What all is happening? We'll go on to irreversible cell injury. What is the mechanism happening there? Then we'll study all these different types of cell deaths in detail one by one. So we'll keep coming back to this flowchart so that you have a better correlation and you have a better outline of the subject of particularly this chapter. So the first thing that we are going to start with is reversible cell injury. And you already know, students, the most common cause of cell injury is hypoxia. So all of this we are going to study. For our simplicity, we'll study it with the help of that what happens when oxygen becomes low. when oxygen concentration goes down. So beginning with reversible cell injury. Now students, please remember, I told you that when there is a decrease in the oxygen concentration, what is the first organelle in our body that is going to respond? Remember, the first organelle affected, that is also an important question that has been asked in the previous year exams, and the first organelle affected is the mitochondria. It is the mitochondria. In fact, students, this chapter is very interesting because over here in this chapter, whenever I ask you that, okay, such and such thing is very common in which organelle, or such and such thing maximally affects which organelle, the answer to most of the questions will be mitochondria. So that is the trick that I'm telling you for this chapter. Whenever I ask you such a question, do keep one possibility of mitochondria in mind. So over here I put up a question that in reversible cell injury, which was the first organelle to be affected? It is going to be mitochondria. Now students, you know that mitochondria causes what? Yes, ever since you started with science in your school days, you were told mitochondria is the powerhouse of the cell. That is, it produces ATP. So how does it produce ATP? It produces ATP by oxidative phosphorylation. Right? But oxidative phosphorylation, look at the term, it needs oxygen. But the oxygen concentration has gone down. If the oxygen concentration has gone down, means there is a decrease in oxidative phosphorylation, correct? If there is a decrease in oxidative phosphorylation, unfortunately, there will be a decrease in the ATP production. So yes, the energy is going to go down. So I hope you're in sync up till now that a decrease in oxygen first affects the mitochondria, as a result of which there is a decrease in ATP. Now let's see what happens beyond this. So now we are going to see all the changes that occur because of this. Number 1. I'm sure you must have heard of something known as sodium-potassium ATPase pump. Sodium-potassium ATPase pump. It has it in its name. It requires ATP. But now I'm saying that the ATP concentration has gone down, which means that this pump is going to undergo failure. In order to understand what is actually happening in such a scenario, you must always know what is actually happening normally in your body. So what I'm going to do is I'm going to draw this particular pump for you over here. So let's consider this to be a sodium-potassium ATPase pump, a cell where this is functioning. And I'm going to draw one line over here so that we can see the normal and the abnormal in the same cell. Okay, now students, what happens normally? Normally, in a sodium-potassium ATPase pump, 3 sodium are going to exit and 2 potassium are going to enter. So this is what is normal, right? What happens? 3 sodium exit and 2 potassium enter, and this in turn requires ATPase. But now I'm saying that we do not have ATP. This pump is going to fail. So what is going to happen? In case of injury, in case of reversible injury, this pump will undergo reversal. So now what will happen? Now we are going to have entry of 3 sodium, complete opposite. 3 sodium will enter and 2 potassium will exit. So there's going to be a reversal. Normally what was happening? 3 sodium exit, 2 potassium enter. The net concentration of ions outside remains more. Why? 3 are exiting, only 2 are entering. Now what is happening? 3 sodium molecules enter and only 2 potassium exit. So the concentration of solutes inside the cell is definitely going to be more. Now when the concentration of solutes inside the cell is more, what will it draw towards it? Yes, it will draw water towards it, right? Because there is an increased hypertonic environment inside the cell, it draws water towards it. Indirectly, I can say that this is going to result in water influx. This will result in water influx. Is this clear? So the first thing you need to know that when there is a decrease in or a failure of the sodium-potassium ATPase pump, the first thing that happens is water enters. I call it water influx. And by common sense, you will answer this for me now. When water enters the cell, what will happen to the size of the cell? Will it increase or will it decrease? You're right. Common sense says that whenever there is water influx, there is going to be cellular swelling. There is going to be cellular swelling. So that is the question that you get. Which is the first morphological change in a cell when undergoing reversible cell injury? This is going to be the first morphological event. First morphological event. This is a question that you've been asked earlier. The first morphological event is cellular swelling for all injuries. For all injuries. But remember, when you go to apoptosis, and when we discuss apoptosis, we will read over there, the cell does not undergo swelling. In fact, there is going to be cell shrinkage. There is going to be cell shrinkage. So, going back to where we started from, there is less of oxygen, because of which the first organelle affected is the mitochondria. So now there's a slight twist over here, students. If the examiner asks you, which is the first morphological event that takes place, you know it is cellular swelling. Correct? This cellular swelling is happening because of the influx of water. So I can safely call it the hydropic change. I can safely call it the hydropic change. Why? Because this swelling of cell is because of water. So if the examiner asks you first morphological event, you know it is cellular swelling. But if the examiner cuts out this word morphological and asks you first event, then remember the first event is going to be a decrease in oxidative phosphorylation. See, the examiner has not asked you the morphology. He's just asked you a general question. What is the first event of this particular injury? What is the first event? So the first event is a decrease in oxidative phosphorylation. But if the examiner particularly says, no, tell me about the cell, what is happening to the cell, what is happening to the morphology of the cell, then your answer will be cellular swelling for all injuries except apoptosis, because over there it is going to be cell shrinkage. Very important questions. Now that you've dealt with this set of questions, I want to take you back to the fact that I told you that because of decrease in ATP production, the sodium-potassium ATPase pump will fail and will stop functioning, because of which we have number one phenomena, that is cellular swelling. Number two, now as the water keeps on entering the cell, gradually there will be endoplasmic reticulum swelling. You know, more and more water is entering the cell, so it will enter the organelles also. Finally comes a time that if this was the cell, and I'm saying that water is constantly entering it, the cell will tend to give up, but still your body is trying to save you, right? So now what the cell forms? The cell ends up forming certain cytoplasmic blebs. There is formation of cytoplasmic blebs. The next thing is that there comes a time that finally damage will begin, because so much of water is entering the cell that damage will start happening. That is when we have formation of something referred to as the myelin figures. We have myelin figures. Now this does fetch at least 2 to 3 type of questions, so let's finish off those. First of all, what is myelin figures? Question number 1. Remember, this is composed of phospholipids and calcium. And please remember both, because students tend to think this is just phospholipids and that's it. But it is actually a combination of phospholipid and calcium. Now students, what I want to tell you over here, phospholipids you already know is from the cell membrane. So once a question came in AIIMS exam that myelin figures are derived from, are they derived from ribosomes, are they derived from peroxisomes, are they derived from lysosomes, or are they derived from the cell membrane? So if you know that, okay, myelin figures was something to do with phospholipid, you will know that this is derived from the cell membrane. So now some lipids from the cell membrane have started to break off and they are combining with calcium. What are they forming? They are forming myelin figures. That is question number 1. Question number 2 says that which injury do we see myelin figures in? So remember, in which injury do they appear? They appear in reversible or irreversible? Correct, you are right. They appear in reversible cell injury. They appear in reversible cell injury, but then comes a twist. They increase more. They are more in number in irreversible cell injury. So it's not that they will come in reversible cell injury and in irreversible cell injury they will disappear. No, that is not the case. They will appear in reversible cell injury, and as the injury progresses into irreversible, their number will increase. Now comes question number 3, and that is the question which is the trend of these days, the trending question, that is going to be the image-based question. So let's see an image of a myelin figure. Now students, till now all that you knew was pathology, something that is all pink and blue, but now I've put across an image which is black and white. Remember, such an image is an image of an electron microscopy. So this is a concept that you go on for now onwards. Whenever you see such a black and white image, we are dealing with which type of microscopy? We are dealing with an electron microscopy. And remember, this electron microscopy, you see this concentric pattern over here. So I want to erase this so that you can appreciate it again. Do you appreciate this concentric layering that is happening? Now this is referred to as the concentric lamellations that you see in myelin figures. These are the concentric lamellations of myelin figures. And what are these concentric lamellations made up of? You're right, they are made up of phospholipid and calcium. So coming back, students, I want you to recall that when there was a sodium potassium ATPase pump failure, a lot of water started entering the cells, as a result of which the first morphological event occurred, that is cellular swelling. With more water entering the cell, we had formation of endoplasmic reticulum swelling. Further more water, we had cytoplasmic blebs. Finally comes a time when damage starts, and that is formation of myelin figures. What is the composition? Phospholipid and calcium. Where is phospholipid coming from? The cell membrane. Which injury does it begin in? It begins in reversible, but the number increases a lot in irreversible. And finally, the concentric lamellations that you need to know, those are important. These are the electron microscopic findings of myelin figures. Coming back, now because of a decrease in ATP, it is not that only the sodium potassium ATPase pump will be affected. You do realize that the body is in a hypoxic state. The oxygen concentration has gone down, which means that we will now begin with anaerobic glycolysis. We will begin with anaerobic glycolysis, right? Now, when we begin with anaerobic glycolysis, students, remember, and I hope you can recall from biochemistry, that there is a very important byproduct known as lactic acid, correct? What do you think will happen to the pH of the body then? You're right, the pH of the body is going to decrease, acidic environment. So because of a lack of oxygen, we have anaerobic glycolysis, because of which we have an acidic environment in the body. On to that, because of this, there is going to be nuclear chromatin clumping. There is going to be nuclear chromatin clumping. So now do you notice in the first part of the pathogenesis, when we were disrupting the sodium potassium ATPase pump, in the end we started breaking the membrane by forming myelin figures. So the membrane was starting to lose up a little. After this, in anaerobic glycolysis, I'm saying that the nucleus starts getting affected. So now the nucleus is also going here and there. So the membrane is also getting affected, the nucleus is also getting affected. Now comes the third hit. The third thing that is going to happen is, I want to draw this diagram out for you first. I hope you can recall this is the endoplasmic reticulum. And now over this, I'm going to draw these structures which are known as the ribosomes. So when the ribosomes are attached on the endoplasmic reticulum, we call it the rough endoplasmic reticulum, and this is what, yes, this is the site for protein synthesis. What happens when injury starts and oxygen decreases and ATP decreases? The ribosomes are going to get detached. The ribosomes get detached. Now, if the ribosomes are going to detach away from the endoplasmic reticulum, will protein synthesis occur? No, you're right. So, there is going to be a decrease in the protein synthesis. There is going to be a decrease in the protein synthesis. So, now if you see, I've slowly and steadily I've told you that the membrane is also getting damaged, the nucleus is also getting damaged, and the protein is also getting damaged. And somewhere you know, these are the three building blocks of a cell: the membrane, the protein, and the nucleus. So the key points over here are that because of a decrease in oxygen, the first organelle that got affected was the mitochondria. So you can say that the first event that took place was a decrease in oxidative phosphorylation. Thus, the powerhouse of the cell is going haywire and now we have a low ATP state. Because of that, the sodium-potassium ATPase pump is affected and a lot of water starts coming in. That gives us our first morphological event, that is hydropic change. With more of that, finally we have formation of myelin figures, which are composed of phospholipids and calcium. Other than this, the nuclear chromatin undergoes clumping because there is acidic environment. And finally, the protein synthesis goes down because the ribosomes are detached. All of this, students, even up till now, your body is so kind to you that even if all of this happens, this is being considered as a reversible mechanism. That is, even up till now, if the injurious agent is removed, we can revert back to normal. But the problem arises is when the injury continues. What are we entering into? We are entering into irreversible cell injury. So as you can see in this, as I told you, we had discussed the main flowchart that when reversible cell injury continues, we are now going to enter into irreversible cell injury, right? So now let's see what is going to happen. So from the same flowchart, I'll say that if all of this continues, if all of this is going to, if the injury continues, what am I entering into? Now I can safely say that I have gone into irreversible cell injury. But then you will ask me, okay, what is the point? How do I know? How have you decided that, okay, now I'm going into irreversible cell injury? So what are the deciding factors? What are the deciding factors? Now there are two deciding factors, students, that I want you to remember. Number one, severe membrane damage. Severe membrane damage. And number two is severe mitochondrial damage. Severe membrane damage. And number two is severe mitochondrial damage. Now this is a very important question that has been asked in the previous year exam, that what is the deciding factor or what is the point at which you say, okay, yes, the cell has gone from reversible to irreversible? That is when the membrane is severely damaged and when the mitochondria is severely damaged. So now with this concept, let's understand what is exactly happening in irreversible cell injury.